Check it out now, y'all.
nanoHUB-U, online instruction.
Hi, and welcome back to Lecture 4 in the last week of Part 2 of Dynamic Atomic Force Microscopy Methods. In the last 3 classes this week, we had been talking about how the atomic force microscope can be used to measure electrostatic forces as well as magnetic forces, and we had talked about some of the issues and complications that need to be taken into account while dealing with those measurements. What we will do today is to change gear a little bit and move towards dynamic atomic force microscopy in liquid environments. It's a good question to ask. Why would you rather put a atomic force microscope in liquid or aqueous environments? And the reasons for that are many, and what you see on the top, for example, is a very important growth area of atomic force microscopy, which is the use of AFM for understanding biological samples. Biological samples, whether it's proteins, DNA, or when you're looking at more complex macromolecular assemblies, such as viruses or subcellular components or protein motors, when you start looking at all these, it's very clear that their functioning in a real biological setting, their shapes, really depend a lot on the presence of water. Biology occurs in water. So to understand the connection between the structure and function of a biological species, it's pretty important to be making those measurements in physiological or near physiological conditions, which typically implies saline solutions, aqueous solutions. So, for example, on the top left, I show an example of a virion. This is a Phe29 bacteriophage that's been imaged in liquids sitting on graphite using a magnetically excited cantilever. And next to it is shown a cryo-electron microscopy reconstruction of the same virus, which is done not in liquids, using electron microscopy at low temperatures to try and figure out what the structure is. And you can tell that there are a couple of interesting differences. You know, the atomic force microscope is able to make out many of the capsid units, the little spherical bumps that you can see actually on the topography of the sample. But it's not able to make them out to as high, apparently, a resolution as what electron microscopy suggests, which when you think about why that is so, the reason becomes somewhat clearer. It has to do with the fact that in atomic force microscopy, you've got a tip, and the tip is going to deconvolve the sharp features of the image a little bit. So you can always expect, first of all, that in atomic force microscopy images, features will be broadened a little bit because of this convolution effect. The second important thing is that when you're imaging in liquids under ambient temperature conditions, many of these proteins that we're looking at are not frozen. They're actually moving around. They're floppy, and you know, the AFM picks out on the flexibility of these molecules as well, which is a pretty realistic situation. On the top, in the middle, on the top row in the middle, you see examples of a image of microtubule in liquid media and in aqueous solutions, and you see, actually, if you look carefully, the linear molecular structure of the chains of the polymeric molecules that make up the hollow tube-like structure that's found within many eukaryotic cells. On the top right, you see images taken of molecular motors with very high resolution. So whenever one wants to study not only the topography of biological samples, but also mechanical properties by doing indentations or other forms of force spectroscopy, the atomic force microscope has an unparalleled advantage over other ways of imaging very small objects. Another growth area where the AFM is becoming useful in liquids is the study of liquid-solid interfaces in and of itself. So there are a lot of interesting physical processes that occur in liquid-solid interfaces that really need to be understood, and really the only mechanism out there, the only instrument out there that allows us to do it with such resolution, both in space and in forces, as the atomic force microscope. So for example, on the bottom right, I'm showing you near atomic resolution images taken of mica, freshly cleaved mica, in buffer solutions. On one hand, you see the topography showing the lattice spacing of the atoms that make up the top surface of mica. On the right, you actually see phase contrast images, which in this case, in this particular paper, have been used to come up with a map of local adhesion on the liquid-solid interface. So problems such as measuring properties such as adhesion or electrochemical properties or diffusion and dissociation of ions on liquid-solid interfaces, all these problems are a very important growth area when it comes to dealing with energy storage, battery-like interfaces, when dealing with wetting and desorption on surfaces. In this whole field, the atomic force microscopy also plays a very important role by studying the properties of the liquid-solid interface itself. Now, before moving ahead with talking about some of the key aspects of what happens when you take an AFM in air and vacuum and put it into liquids, it's worthwhile, posting a caveat here, which is that unlike air or vacuum, and in some sense in a very different way from air and vacuum, preparation of samples for liquid environment imaging requires a lot of attention to sample preparation. In particular, sample preparation requires a very good understanding of surface functionalization and surface forces to ensure that your biological sample remains adhered to the substrate and doesn't float up when immersed in liquid. And there are many specialized techniques that have been developed for trying to immobilize biological samples on surfaces. And I encourage you that if you haven't tried atomic force microscopy in liquids, that paying a lot of detailed attention to sample preparation protocols is essential for liquid environment AFM. Okay, so what happens when you take an AFM cantilever and put it into liquids? The first thing that I'd like to bring to your attention that changes is the nature of tip-sample interaction forces. In part one of the class, you talked about a variety of interaction forces which are applicable to air or vacuum situations, but you did not have a chance to consider what happens in liquid or aqueous saline solutions. So that's what we'll begin with in this particular lecture, is to get an overview and understanding of what are the kind of forces that exist in liquids. Now, broadly speaking, the forces that are important when dealing with dynamic AFM in liquids are listed here. We're going to focus today on the electrostatic and van der Waals forces through what's called the DLVO theory. We'll talk briefly about hydration forces, hydrophobic interactions, and steric forces, all of which also play an important role. So let's begin with talking about electrostatic and van der Waals forces. So what I've drawn here is a sketch of showing a progression of events that occur as a tip is immersed in a sample. So what we have is the sphere. The sphere represents, let's say, a sphere of radius r sub tip, which is approximately the radius of the end of the AFM tip. And there's a sample, and it's immersed in a solution that typically is water that contains dissolved salts in it. The salts can be monovalent, like sodium chloride, potassium chloride, or they can be bivalent, like magnesium chloride, or trivalent, etc. Now, one of the first things that happens when you take a tip and put it into such a saline media, and to put a sample into such a saline media, is that charge starts accumulating on the surface, on these surfaces. The reasons for this are actually pretty complicated. one of the main reasons why charge develops on these surfaces is because of the fact that there is an absorption at the surface of ions from the surface. There's a differential absorption. So for example, depending on the chemistry of the surface, it might actually absorb only positive ions from the surface or only negative ions from the surface, depending on the interaction with the surrounding media. So that's one important reason. A second important reason is the fact that, the tip itself might have some native charges present on it. If it's an insulating tip and there's some charges, you may also have contact potential of the tip, or you may apply actually a voltage to the tip as well. Nonetheless, the key thing that happens when you put a tip and a sample into surfaces, you end up getting a distribution of charge on the surface of both the tip and the sample. Let us denote the charge density that develops on the tip as sigma T. This is Coulomb per meter square. And sigma sub S is the charge density that develops on the surface of the sample, again in coulombs per meter square. Now, the third step that happens as you move to the right is that the water, being a polar media, the dissolved salt have dissociated into positive and negative ions as well. What ends up happening is, because these surfaces are now charged, they will attract ions of the opposite charge to them. So if you have, for example, positive charge adsorbed on the surface, it attracts negative charge from the solution to start clustering around the positive charges on the surface. And as you can imagine, it's not going to be a single layer of charge that's attracted. There's going to be like a cloud of charges that gets attracted on both the surface of the tip and the sample. And what these do is they create what's called an electric double layer. What this means is there's a cloud of oppositely charged ions that surround the tip, another cloud that sits on the surface. Now, what happens when you move the tip close to the surface is these two electric double layers start interacting with each other. The ions are being pushed out, and the energy required to rearrange the ions in this cloud of layers it results in a force. You have to apply a force to push those ions out. And that is the source of electrostatic forces between a tip and a sample in liquids. It's not the standard electrostatic force argument that was presented in part 1 of the class, and it really has to do with this electric double layer and about pushing these ions, the ions that tend to screen the charges on the surface, trying to push them apart as you bring the tip and the surface close together. Now, one key feature of these forces between the tip and the sample, what I call the electric double layer forces, is the fact that they decay exponentially with separation between the tip and the sample. And the decay length is called the Debye length of the solution. And the Debye length of the solution, at least for a monovalent, when you have a monovalent salt dissolved in water, is given by the expression on the bottom left. The Debye length can be anywhere from a few angstroms to all the way to a micron, depending on the concentration of the salt species in the water. So as you can see that the concentration in moles per liter sits in the denominator. So as the concentration becomes small, the Debye length becomes large. On the other hand, when the salt concentration becomes large, the Debye length can become very small as well. So that's one very important thing. And again, there's a reference shown in the very bottom. This is one of the earliest works that started looking at electrostatic forces and screening effects in liquids. So the question now is, this is great. We now have this concept of Debye length. The question is, how does one calculate the force between a spherical tip and a flat surface in a medium whose Debye length is given by a formula like this? I would like to add that there is a variant of this formula for Debye length for solutions that are not monovalent or do not contain monovalent ions, and that's given in the appendix. All right, so when you're dealing with trying to understand the forces between the tip and the sample in such an ionic medium, there is a very commonly used continuum approach, which is called the Poisson-Boltzmann equation, and it's really the solution of the Poisson-Boltzmann equation that is able to tell us what the force is between the tip and the sample as a function of distance. However, when solving the Poisson-Boltzmann equation, which is a partial differential equation, one needs to supply some boundary conditions to it. And there are generally two kinds of boundary conditions you could apply. You could say that the tip and the sample are at certain potentials or voltages, and those voltages do not change as you come close to the sample. Ideally, that would be the case if you're dealing with a metallic tip, metallic sample with no contaminants on top, and that you're controlling the voltage potential applied to those and you bring them close together. That would be a situation where you're fixing the potential on the two surfaces, solving for the Poisson-Boltzmann equation, and from that computing the pressure and the force between the tip and the resultant force between the tip and the sample. The other boundary condition you could apply is to say that, no, to recognize that in many cases we actually have dielectric tips or insulating tips and insulating samples that have charge that develops on them by dint of being in the saline solution, as just explained on the previous slide. So that in that case, what one would do is to assume that we know a certain charge density on the tip, sigma t, that we know a certain charge density on the sample, sigma s, and that those charge densities we assume do not change as you bring the tip and the sample together. We would also have to exploit the spherical symmetry of the problem to solve the Poisson-Boltzmann equation. But it can be shown that the solution of the electrostatic force turns out to be, for these two cases, as shown in equations 2 and 3 here. The first equation is for the case where you have given or provided constant potentials, and the second case is for when we have or we know the value of the surface charge in the tip and the sample. What's interesting about both these expressions is that, first of all, they're slightly different from each other. So the constant potential case and the constant charge density case actually don't give the same expressions. Their dependence on, for example, the Debye length is different. The other thing is that when one takes the limiting case of large distances, if distance become very large, we actually do find that the constant potential and the constant charge case actually tend to asymptote to each other. So they're not too bad. You can use either one if the tip sample separation distances are much larger than the Debye length. Nonetheless, it's important to understand the validity of these equations shown here. They're valid for whenever the tip radius is much larger than the Debye length, and they're also valid for when the separation is more than the Debye length itself. Now, in addition to the electrostatic forces, we have to worry about van der Waals forces, which also exist in liquids. And we've already studied the general form of van der Waals forces between a sphere and a flat surface in part 1 of the class. We're going to use the same result as done in part 1 of the class. In part 1 of the class, you actually derived the expression for the potential energy associated with van der Waals forces between a sphere and a sample. The corresponding force is simply negative of the gradient of that potential energy. What one gets out of this is that the van der Waals forces are attractive, so there's a negative sign. H is the Hallmarker constant, R tip is the radius of the tip, and it's divided by 6 times d squared, where d is again the separation between the tip and the sample. So the DLVO model for tip-sample interactions assumes that the net forces between the tip and the sample before the tip interacts with the sample, so we're talking about non-contact interactions, arises due to 2 effects. It arises due to electrostatics plus an effect due to van der Waals forces. Now, we do know that the van der Waals forces are short-range forces. They typically act at distances, up to 1 to 2 nanometers max. The electrostatics forces, on the other hand, are controlled by the concentration of the salt in the solution by the Debye length. And as I said, those can be as short as 2 to 3 nanometers, but they can be Debye length can be as long as a micron, for example, in deionized water. Again, this combination of van der Waals forces and electrostatic forces is really the essence of what's called the DLVO theory. That's based on a couple of papers in the 1940s that really revolutionized the field of colloidal stability. What had happened, as I described to you, Debye and Huckel had first worked out the whole idea of electrostatics in liquids, but the application of that idea to and the addition of van der Waals forces to study the stability of colloids it was really in the 1940s, took place in the 1940s, and this theory, DLVO theory, pretty much was the basis of understanding why is it that when you take a colloidal solution and you start increasing the concentration of salt in it, why do very often particles coagulate and come together, and why you need low salt concentrations for separating. And the answer to it has to do with the following observation, that as you decrease salt concentration, the electrostatic forces act over a much longer range, and the Debye length is large, and electrostatic forces are generally repulsive, keeping the particles apart. On the other hand, when the Debye length decreases by increasing salt concentration, the repulsion occurs only at very short distances, by which time the van der Waals, or at which distance the van der Waals forces become active, and they can suck the particles together. So this whole notion of DLVO forces was first developed really for understanding colloidal stability, but it's fortunate that we can use the same ideas to understand AFM in liquids. All right, so let's put some questions up here. These models that I just showed you before provide good predictions as long as we're dealing with distances, as I said, greater than Debye length, as long as we're dealing with moderate surface charges and moderate potentials. But the big question is, which of those two formulas would you use in your particular case, equations 2 or 3? And that is one of the challenges that comes into applying the theory, that it's very, very rarely the case that you know exactly which boundary condition you need to apply, unless you've taken a lot of effort to make sure that the two surfaces are conducting, and you know there is no contamination on that, and that you can apply known potentials, for example. So that's one challenge that comes up. Now, if you think you're going to be dealing with insulating tips, which is very often the case, for example, in liquids, you might want to deal with silicon nitride cantilevers or silicon oxide coated cantilevers, and you might deal with samples like mica or biological species, which are also insulating. In that case, you need to know what the surface charge density is on the tip in the sample to really apply this formula. And there also a complication occurs in that the surface charge density actually ends up depending on the pH of the solution. Depending on the pH of the solution, you can actually have different kinds of charge densities on a surface. And so, in fact, there is a pH value, at which you can actually get the charge density to be nearly zero as well for a given surface. So the key thing is that the quantities that make up the different components of this interaction force depend on a number of parameters, and we need to take that into account. All right, so what you see now below is an application of equation 3 from the previous page, which has been worked out for a silicon nitride cantilever tip as a function of distance for different salt concentrations for a monovalent salt. So I want to bring to your attention 3 interesting features that come up in these graphs. The first is that when the concentration is very low, salt concentration is very low, you can see that the electrostatic forces extend over a much longer range. They start acting at much longer ranges. And as you come closer, you can see a bit of the effect of the attractive van der Waals gradients that starts coming in. So when you're dealing with low salt concentrations, what's interesting is that van der Waals forces can be positive, can be net, the net forces can actually be positive, but the van der Waals forces can actually cause a negative gradient to occur. So whereas the total force still remains positive, but you can get a negative gradient, slight negative gradient at low salt concentrations due to the effect of the van der Waals forces. On the other hand, as you start increasing the salt concentration, two things happen. The Debye length decreases, but also the intensity of the electrostatic forces starts to change, so much so that the van der Waals forces start getting screened very strongly. So that when you deal with very large concentrations, you're dealing with effectively no interactions until you come very close to the surface, and then suddenly you start, and then you get this exponential growth due to the electrostatic forces, and the van der Waals forces are not very visible. So what this result brings to our attention are two important implications that come out of this. The first is that van der Waals forces or adhesive forces in liquids are going to be very diminished compared to air, because they are opposed by this repulsive electrostatic fields, repulsive forces that develop between the tip and the sample, offsetting the negative forces from the van der Waals and very often completely screening them as the salt concentration increases. This has very important consequences. One important consequence is that one can use very soft cantilevers in liquid environments. You can use cantilevers as soft as 0.01 Newton per meter. So you can use cantilevers about 100 times softer or lower stiffness compared to that you can use in air, precisely because there's very little adhesion and the tips don't tend to snap into the sample. They can if you use very small cantilevers or very small stiffnesses, but very often the adhesive forces are much smaller compared to air. So that's one important conclusion that comes out of the study as well. I'd like to bring to your attention on the next slide some examples of the force distance curves that you might get in liquids. On the top is work out of a thesis, Javier Sorthuris, whose advisor is Arturo Baró. And what you see is an example of a force distance curve, force Z curve, on a mica substrate in a PBS solution using a very soft cantilever. And you actually find that as you approach the sample, the cantilever bends up due to repulsive forces, but then snaps in due to the attractive van der Waals forces as you come further in, and there's a snap out corresponding to the van der Waals forces, but then after that it follows the electrostatic repulsion line. And if you convert the FZ curve to FD curves, as you've learned how to do in part 1 of the class, you see a very clear reconstruction of the DLVO forces, which show a repulsive, exponentially growing electrostatic force, followed by a little dip that comes from the van der Waals forces. On the bottom left, this is work out of Daniel Miller's group, and it shows that the force versus distance graphs, force versus Z graphs, how they change with salt concentration. And as you can see, at very low salt concentration, you have long-range electrostatic forces that grow exponentially, but as you decrease the salt concentration, everything gets concentrated in a very small region of interaction between the tip and the substrate. And you pretty much then get a very sharp transition from the zero force to the repulsion due to contact. On the bottom right is also an interesting result that shows how the force distance curves acquired between a tip and a sample are affected by the pH of the solution. Remember, I mentioned to you that the pH of the solution actually influences the charge density on the tip and the sample. As a result, the electrostatic forces also can change significantly. So that gives us an idea of DLVO forces between the tip and the sample. There are a bunch of other forces which we have not talked about so far. Probably the most important one of them is the hydration force. The hydration force refers to the force, which is a very short-range force, typically happens in about a nanometer or two between the tip and the sample. The DLVO forces were all based on an understanding of continuum electrostatics, but when you come to such small spacings, you cannot deal with water as a continuum. You have to worry about it as a layered medium of molecules and so on, layered molecules of water. So the hydration force refers to the force that's needed to remove the water that's naturally associated or layered on surfaces in solvation like shells. So as you bring a tip close to the sample, you got to imagine that there's a few layers of molecules that are adhering or they're hydrated to the tip in the sample. As you bring a tip close to the sample, those have to be pushed apart. So that's a short-range repulsive interaction, often modeled by an exponential term. In the case of molecularly flat surfaces, people have observed that the hydration forces are actually oscillatory. But that's an important force that comes in for really short-range interactions. In addition, another important force is a hydrophobic interaction force. And this is one of those forces that's still poorly understood. But when you're dealing with hydrophobic tips and samples, basically the hydrophobicity of the surfaces makes it so that the tip in the sample wants to remove the water from a substrate. And what ends up happening physically is when you're dealing with hydrophobic surface in water, you actually end up seeing a lot more adhesion between the tip and the sample. This is because the tip and the sample prefer to be attached to each other rather than having water in between them. And this is a short-range chemical force that leads to increased adhesion between the tip and the sample. And there has been evidence in the literature, as shown at the bottom here, that hydrophobic surfaces are also associated with the creation of nanobubbles on hydrophobic surfaces. The last kind of force that could be useful or relevant in liquid settings is steric forces. This force refers to whenever you're dealing with AFM tips or sampled with grafted long molecular chains hanging from it. Again, the details we cannot go to of the origin of the steric forces in this particular class, but there's a reference at the bottom here which will suggest other references where you can go and study this at greater depth. So thank you so much, and we'll see you in the next class.